Hi there, I'm Susan Lindquist. I'm at the Whitehead Institute at MIT and in the Howard Hughes Medical Institute. And I'm here to tell you about protein folding and how it can act as a very powerful driver for the evolution of novelty in a variety of different systems, in a variety of different ways. I'm going to tell you about our work in two parts. The first part will be about HSP90, a very special protein that helps other proteins to fold and has multiple ramifications in terms of the evolution of novelty. So I'm going to start perhaps by talking just a bit about this amazing man here, Dobzhansky. In the 1930s, he really realized that within the context of Darwinian evolution, we really needed to have a new synthesis that took advantage of some of the new insights in biology that were emerging at that time. And he really made a very coherent, comprehensive revamping of our understanding of evolution, had a tremendous influence on the field. And this is one of his most often quoted statements: nothing in biology makes sense except in the light of evolution. And that's absolutely true, and it's a mantra just about every biologist I know lives and breathes by. But he also, interestingly, said this about the possibility, the Lamarckian, and initially envisioned Lamarckian possibility, that inheritance of traits could be done in a rather different way from what Darwin had originally envisioned, and that would be that the environment could influence the appearance of a new trait, and that could then be passed on in subsequent generations. And about that, Dobzhansky was not too wild about that concept. And he said this question has been discussed almost ad nauseum in the old literature, so that we may refrain from the discussion of it altogether. But what I'm going to tell you about today and in the next lecture is the fact that actually our understanding of how biological systems work, and particularly how protein folding influences those biological systems, has actually provided a perfectly rational and reasonable explanation for how the environment can lead to the appearance of new traits in evolution. And it actually fits very nicely within a Darwinian network of selection and mutation and variation. So let me tell you how I got into this. We were working on something called the heat shock response. And this is it in a nutshell, envisioned in three very, very different organisms, very, very highly conserved process. So what we have here are yeast cells on the far end there, and Arabidopsis seedlings, and human tissue culture cells. And in each case, they were started out identical, but the top row was treated immediately at a high temperature, which killed them, as you can see. The bottom row, those cells or plants, were first exposed to a moderate, very mild preconditioning heat treatment for a short period of time. For example, for the yeast, it was just half an hour at 39 degrees, warm summer day temperature. And that allowed those cells to acquire an extraordinary ability to survive much more extreme conditions. And as you can see, the same thing applies for the plants and applies for the tissue culture cells. It applies to every organism on the planet. Birds do it, bees do it, even educated fleas do it. Every organism responds to moderate stresses by adjusting to the possibility that there might be much worse stress coming along and getting ready for it. So how do they get ready for it? The way they do it is actually to make a variety of proteins called heat shock proteins. So these are revealed by simple radiolabeling of cells at the normal temperatures. If this is yeast cells, normal temperatures of 25 degrees versus that 39-degree conditioning treatment. And you can see that what the cells are doing is they've started to make a whole bunch of new proteins. These proteins have been visualized by gel electrophoresis, and they've been spread out according to their molecular weight. And they're named heat shock protein, HSP 104, 90, 70, and 60, just simply according to the sizes of the protein. Now, it turns out that these are induced by many different forms of stress. They were first found in response to heat, and that's probably a very, very common universal induction in nature. But they're induced by all kinds of stressful conditions, anaerobic conditions, altered pHs, changes in carbon metabolism and food sources and energy, etc. And they provide protection against an enormous number of different stresses. The proteins are very, very highly conserved, such that you find these same proteins being expressed not only in the organisms I've shown you, which are all higher organisms, but even going out way back to the deepest lineages of organisms, the prokaryotic organisms known as eubacteria and archaebacteria have heat shock proteins as well. And it turns out that all of these proteins that are produced in massive quantities and very quickly in a kind of an emergency response to stress, all of them have to do with the protein folding problem. So what is the protein folding problem? Well, this is an example of one protein. The DNA code is long and linear, and it has to fold into this very, very exact, precise shape. This protein, when it's decoded from that linear DNA, has to fold into a very precise shape to function. And each protein folds into a completely different shape. And they do that in a just kind of crazy environment. Cells are absolutely crowded. And so those proteins have to come out of the ribosome in long linear strings and fold up into the right shape with the presence of a whole lot of other proteins. And the thing about this that's really quite extraordinary is the kinetic energy, because those proteins aren't just sitting there as they are in this static picture. They're moving around like crazy. They're banging into each other at an incredible rate. And that means that it's hard for them to get folded in the first place, and it's hard for them to keep those folds. All organisms face this problem. Life is driven by the proteins that are coded by DNA, and all of those proteins have to fold. They fold in a chaotic environment, and stresses make that environment even more chaotic. So it makes it more likely that those proteins will start misfolding, and that's why they synthesize all these new heat shock proteins, whose sole function is really to help those other proteins stay folded, keep folded, and not get into aggregated messes. So what I'm going to be telling you about today is about one particular member of the heat shock protein family, and it's called HSP90. It's a particularly interesting member of this group of proteins for a variety of reasons. One is that it's always far more abundant than is needed under normal circumstances. Every eukaryotic organism on the planet, at least that is going from yeast to us, is making much more of this protein than it really needs under normal circumstances. And that's actually not true for some of the most of the other heat shock proteins. Most of those are made at just the levels that they're needed and no more. So that means that this excess HSP90, which is one of these protein folding aids, is there to serve as a buffer against environmental stress and a buffer against the protein folding problems that occur with environmental stress. And the other aspect of HSP90 that's quite special is that it has a tendency to have a more restricted group of proteins that it helps. It doesn't help every protein in the cell. It particularly helps proteins that are known as signal transducers. It helps others too, but the main ones that it seems to concentrate on are signal transducers. Signal transducers are proteins that respond to just about everything in the cell. They respond to developmental clues, they respond to hormones, they respond to stresses in the environment, but they are what really drives the biology of virtually every organism. And so they're the major regulators. There may be long pathways involving lots and lots of proteins, but these signal transducers drive whether that pathway will be on or not on under different circumstances. So they're very dynamic, and they're key to regulation of other cellular processes. So together, the fact that there's this excess protein folding buffer made up of HSP90, and the fact that that buffer is specifically directed towards these really dynamic, really important central regulators of growth and development, creates a really interesting situation for evolution. It allows HSP90 to act as both a potentiator and as a capacitor for vast amounts of genetic variation. And we found this to be true in every one of those organisms I've just shown you, from yeast cells to Arabidopsis seedlings to human cells, and and in fact, many other organisms in between. So by acting as a potentiator and a capacitor for so much genetic variation, and it's not that other proteins aren't acting as to help other genetic variants exert phenotypes. This one just is a particularly powerful driver of, of the effects of genetic variation. And that facilitates the appearance of really complicated traits. One of the problems in evolutionary biology is how can organisms rapidly evolve what seem to be really quite complicated traits? And there are many cases in evolution where we know this has happened. Evolution isn't always a slow, gradual process. And it's been hard to envision how that could happen if each individual mutation that contributes to that trait has to occur independently and then be selected for and not make things untoward for the organism. HSP90 provides a way in which lots of variation can be either potentiated or buffered all at the same time. And that causes that variation to change with environmental stress, because environmental stress needs all other proteins start to need HSP90. They take away that buffer, and then you see the effects of the variation in other parts of the genome. So that's the basic story I want to tell you. So let me illustrate with several different kinds of examples. So one of the first proteins we looked at, and in fact the first one that gave us a hint about the power of this protein in terms of evolution, was a protein called VSARC. VSARC is actually the first oncogenic protein found. That is the first cancer-causing protein found. And it's a mutant version of the normal cellular protein called CSARC. Well, we've been studying this heat shock response for a variety of other reasons. And we realized that it's so highly conserved. And so we realized that maybe people had found VSARC associated with HSP90, and they didn't really know quite what it was doing associated with HSP90. They'd found it associated with HSP90, but of course this was in human cells and rat cells and higher organisms that are prone to cancers. But because this whole system of heat shock proteins is so highly conserved, we thought, well, maybe we could test what that relationship between HSP90 and this cancer-causing protein was by using the yeast cells as a sort of a living test tube. They have all the crowding and all that protein quality control issues that our higher organisms have, but they're really simple and easy to use, easy to work with, and manipulate genetically. So we took this cancer-causing protein and we put it into our simple little yeast cells. And the reason why we did that was that we could make yeast cells genetically very easily that had normal, high, lots of abundant HSP90 or lower levels of HSP90. That was the lower levels that could take care of all the normal functions needed. The cells couldn't grow at higher temperatures, but they were fine at normal temperatures. And just see, well, what was the difference in the behavior of this protein that associated with HSP90 when there was a difference in the level of HSP90? It actually had previously been thought that the association between this protein and HSP90 was keeping that kinase inactive, because in the complex with HSP90, when it was purified, and this was done with several different kinases actually in several different laboratories, that oncogenic protein, which is a kinase and modifies other proteins to cause growth, it was inactive. So naturally it was assumed that HSP90 was inhibiting it. So we tested that. In our yeast cells, which had low levels of HSP90, if HSP90 was inhibiting that protein, now it should become much more active. And we got exactly the opposite result. So first you can see on the left over there that we've got simply looking at the level of SARC expression that we've engineered into these yeast cells. And with our wild type cells or the cells that have reduced levels of HSP90, it's pretty simple. It's pretty similar. There's not much difference between them. But when we assayed the oncogenic activity of that kinase, its ability to modify other proteins with phosphate and use those phosphate modifications to change the biology of the cell, and in human cells fuel cancer and malignancy, it was when we reduced HSP90, the activity of that protein didn't increase. It was completely dead as a doornail. And the evolutionary aspect of this became clear when we said, well, wait a second, let's look at the normal cellular kinase. And there we had a really interesting result. So when we put the cellular version of the kinase into yeast cells and we monitored activity, now we had to expose this blot for about 20 times longer than we had exposed the oncogenic cancer-causing blot because the normal protein is, of course, much less active. But when we exposed it 20-fold longer and we were actually able to see the activity under the two different circumstances, wild type levels or low levels of HSP90, there wasn't much of a difference. There's a little bit of a difference, but not much. So that indicated that HSP90 was particularly important. This buffer, this excess amount of HSP90 was particularly important for that cancer-causing protein, the newly mutated protein, to be active. So is this just an oddity of yeast cells? No, it turns out that someone who has been a wonderful colleague and friend of mine over many, many years, Luke Whitesell, did a very, very interesting experiment in mammalian cells. So he found that gildanomycin, an HSP90 inhibitor, reverses the effects of this cancer-causing protein in human cells. So here are cells that are the control cells are growing out of control because they're expressing this cancer-causing protein that's causing them to grow too much, and they're just kind of piling up all over the place, out of control. When the cells are exposed to gildanomycin, which brings down that HSP90 buffer and inhibits it, they slow down, they spread out, they get contact inhibited, they're normal. The interesting thing about this experiment was that it was initially not realized until Luke did some really interesting biochemistry that's what he was doing with this gildanomycin. When he first bought this, and this is an interesting example of how science keeps turning things upside down. When he first bought gildanomycin, it was advertised in the Sigma catalog as a Src inhibitor. And that's why he had applied it to these cells, because he wanted to see whether inhibition of Src would reverse the cancer phenotype. But when he purified Src and added the compound and looked at its kinase activity on a gel, there was absolutely no difference. That compound was not inhibiting the kinase. So he put the compound on a column that allowed us to fish out the proteins that compound was binding to. And as you can see here on this gel, there's one protein that compound's binding to, HSP90. First inhibitor of HSP90 found, and it allowed this test then of is HSP90 buffer serving that same function in these human cells. If you reduce that excess amount of protein, does it prevent that new phenotype that was caused by that new mutation? So with a long series of experiments by several other laboratories, we now, and especially want to point out Johannes Buchner, by the way. This is a not easy biochemistry to do, and Johannes Buchner's group did a lot of it. The picture we have is now much, much clearer. So Src is a protein that sits in the cellular membrane, and it normally folds back on itself and keeps itself auto-inhibited. And it only signals for cells to grow every once in a while under normal conditions when they're supposed to grow. What happens in those cancer-causing mutations is that the protein can no longer fold properly because that part that tucks into itself and keeps it folded back on itself and inhibited is no longer working. So the protein unfolds. It's very, very unstable, and it would just normally get degraded or aggregate in the cell and not be good for anything. HSP90, however, sees it as a protein that's in trouble folding. It comes in, it swoops in and says, oh, you're just the kind of protein I'd like to bind to, a signal transducer that activates cellular pathways, so I'm going to help you fold. So it helps the protein fold, helps get it to the membrane, and keeps it active. So HSP90, in this case, this excess buffer, has been absolutely essential for that new mutation to create this very new phenotype, the oncogenic state. That has turned out to be true for many, many other kinases now that cause cancer. So it's a very broad truth in the cancer kinase literature that HSP90 is important. And because it's just, you can inhibit that oncogenic transformation by just reducing the buffer, not eliminating the protein, but just reducing the buffer so that it doesn't hurt other proteins in the cell. The idea has been that this might be a very useful clinical tool in a variety of different cancers, and that's being tested by many pharmaceutical companies now. So what about a completely different kind of evolutionary process? We turned here to a very different group of organisms. We turned to fungi. And but we also wanted to study something that was maybe important to human biology and medicine, and for which there were because it was important in human biology and medicine, there were a lot of tools that we could take advantage of in terms of studying evolutionary processes. So we chose to study the evolution of drug resistance in fungi, because fungal infections are a horrific problem. These take a variety of different forms, but when fungal infections become systemic, when they've really invaded us, there is very little that we can do about it, and the mortality rates are 40 to 90%, depending upon the fungus and the circumstances. The reason why the mortality rates are so high is there actually are very few antifungal agents that we have. There are really only 3 major ones that are deployed in the clinic right now. The reason for that is very interesting. Even though you would think of fungi and human beings as being very, very unrelated to each other, it turns out that their biology, if you look at the evolutionary tree, the eukaryotic lineage, which split off from Archaebacteria and Eubacteria a long, long time ago, and then there was a whole lot of evolution took place before fungi, plants, and animals split off from each other. And so there haven't been very many selective targets. There's a lot of antibiotics that target things about bacteria that have nothing to do with a human being. Haven't been that many successful ways of targeting things that were specific to fungi that are not found in human beings. So resistance is evolving. It's becoming a big problem, and we thought it was worth studying. So here, what we've done is a very simple experiment. We've taken Candida albicans. We did this actually with the lab rat yeast, Saccharomyces cerevisiae, first, because genetically you can manipulate it much, much better. Candida albicans is a pain in the neck, actually, to manipulate. But it's a much more important human pathogen. So what we've done here is we've simply plated it out into Petri dishes. And you can see the cells are growing like crazy over here. And then we've put in this antifungal drug called fluconazole over here, and you can see that most of the cells have died, but not all of them. And in fact, you can start to see colonies growing stronger and stronger and stronger that are evolving resistance to that drug. So we asked, does this kind of resistance that's evolving in the laboratory, does that new phenotype depend upon this HSP90 buffer? So we took advantage of some of these compounds, like the one I just told you about, jaldenomycin and ridicicol, compounds that very selectively inhibit HSP90. And we asked, well, what if we just put in a smallish amount of those compounds such that we took down that excess buffer of HSP90 protein folding capacity? What would happen to the evolution of drug resistance? And what you can see here is that we're using a concentration of that HSP90 inhibitor that has no effect on the normal cells under normal circumstances. They're just growing perfectly happily because they don't need very much HSP90 function for normal growth, but they need it to evolve drug resistance. Now, there are some mechanisms by which they can evolve drug resistance that don't depend upon HSP90 in the laboratory, but an awful lot of them, different mechanisms, do depend upon HSP90, that protein folding buffer, to allow these new mutations to create their new phenotypes. So we got exactly the same results with 2 completely structurally unrelated inhibitors, which had in common the fact that they both inhibited HSP90. And so that when that was a really important aspect of the chemical proof, the experimental way of working with this organism that was so difficult to work with genetically, was to take these two very different drugs and find the same effect. We then later, subsequently, were able to do it genetically, but that took a couple of years. Anyway, this is a laboratory experiment, and evolution doesn't take place in the laboratory. What about evolution in a real life situation? So in this case, we took advantage of some colleagues' wonderful work that had isolated strains of Candida albicans from patients who were suffering from fungal infections. And the particular series of strains that I'm showing you over here is a series of strains that, well, the top one is the laboratory strain. And each row below that is a clinical isolate taken out of a patient over the course of about 2 years. Now, the reason why this is an evolutionary experiment is when the patient got better and then got sick again, it turned out that it wasn't some new fungus invading that person from the environment, but rather the fungus inside that patient had gotten under control from the drugs, but then it rebloomed. And so it went through this series of being under control, blooming, under control, blooming. So it's an evolutionary series. So that's what we call in vivo selection. This is a natural case in which the cells are evolving. Now, you can see the green means growth and black means no growth, and you can see that under all of the different concentrations of fluconazole here, these cells are actually doing pretty well. So even the initial isolate from the patient was fairly resistant. You can see it's getting a little greener down there. It means that those cells are growing even better in the presence of the drug. Did this naturally occurring evolution of a new trait in the human being, the natural, real life evolutionary experiment, did the acquisition of that new trait, drug resistance, depend also upon that HSP90 protein folding buffer to enable those new mutations that caused resistance to create this novel phenotype of drug resistance? Again, we used those inhibitors, and we just brought down the buffer, HSP90 buffer, a little bit. And what we found was that in the absence of fluconazole, the buffer, we're not inhibiting HSP90 all that much, because in the absence of fluconazole, the cells are growing just perfectly fine. But as this concentration of fluconazole is increased here, you can see that the eliminating that HSP90 buffer has completely done away with their resistance. Their resistance depended upon excess protein folding capacity. Now, the other thing that was interesting here was that you can see that as time went on, the cells evolved very robust resistance that was independent of HSP90. Towards the end of this person's life, that organism had become so virulent that and had acquired new mutations, and it no longer was sensitive to the inhibition of HSP90. So what could have driven that? Well, remember what drove that evolutionary process, such that something that was originally a trait that was originally dependent upon HSP90 then became, over the course of time, independent of it, became fixed in effect. Well, remember that these heat shock proteins are taking care of environmental stresses and the protein folding problems that occur with environmental stresses. So it occurred to us that fever temperatures, I mean, we naturally are always mounting fevers in response to infections. Maybe one of the reasons for that is those fever temperatures create protein folding stresses in the organism. That might use up that HSP90 buffer and also take away the drug resistance. And sure enough, that's what happens at least when we look at those strains and examine them in the laboratory. These do not have the HSP90 inhibitor. There's something growing at higher temperatures. And those higher temperatures, we think, are what contributes to the selective pressure over the course of this patient's terrible infection history to create a driving force for new mutations to now allow that resistance to be maintained even when HSP90 functions and other protein folding homeostasis functions are stressed. It's turned out we've now studied this same process in fungi that have diverged from each other by 1 billion years of evolution, that are very, very, very different organisms. They're as different from each other as they are from us, to be honest. And we've looked at it in response to 3 very different kinds of antifungals that cause the evolution of resistance. And we found basically the same thing. When we look at strains that have naturally evolved resistance against these drugs in human patients, and then ask whether that evolution depended upon the HSP90 buffer, found that about 50% of the strains that is indeed the case. So that's a lot of evolution covering a lot of territory. So those are 2 examples of HSP90 acting as a potentiator for new mutations, allowing this protein folding buffer, allowing new mutations to create new phenotypes. And these new phenotypes are lost transiently with stress, and additional mutations that can accumulate, however, can lead to their fixation. There are, it turns out, multiple mechanisms involved. I showed you one mechanism by which the HSP90 buffer, that protein folding agent, helps to drive the phenotype of an oncogenic kinase. It turns out that for these other drugs, the nature of the mutations and the nature of the ways in which they create resistance are quite varied, very diverse, and HSP90 acts in those pathways in a variety of different ways. But it is a really powerful driver of this evolutionary novelty. And that you can look at some of my papers if you want to read the details. I don't want to go into any of those details now, because I want to turn to something else. I want to turn to the fact that HSP90 can also serve as a capacitor for genetic variation. And when it serves as a capacitor for genetic variation, what it's doing is kind of exactly the opposite. Having this excess protein folding buffer allows lots of polymorphisms and genetic variants to accumulate without any effect on the organism. You don't notice them. But then when stress, environmental stress or internal stress, causes problems with protein homeostasis and starts to use up that HSP90 buffer, wow, the effects of that genetic variation now become very important. And so in those cases, those are traits that appear under stress. So quite different from the way in which things behave with respect to potentiation by HSP90. So let me maybe show you, instead of just starting with the results that we got, which initially were a little bit baffling to us, let me kind of place it in the context of a much broader literature. This is a signal transduction diagram of a cell, of a cancer cell, in fact. And signal transduction diagrams are really like circuit diagrams in a computer chip. And they direct the flow of proteins around the cell and the behavior and activity of proteins around the cell. And you can see that, for example, here a growth factor is coming in, it's binding to a protein, and it's sending all kinds of signals along the cell and causing all kinds of things to happen. The arrows indicate the key signal transduction steps, the key proteins that depend upon HSP90. So HSP90 is helping to drive, under its normal circumstances, HSP90 is helping to drive all of these pathways forward. And so you can imagine that if something happens to inhibit HSP90, and that could happen either pharmacologically or just environmental stress is using up that HSP90 buffer and not making as much of it available anymore, those pathways might not work quite as robustly and might not go quite as robustly. And for that reason, genetic variation that was in those pathways that wasn't having an effect previously, because those pathways aren't working so well anymore, those variants can have exert all kinds of effects and cause all kinds of new phenotypes. So how did we get there? Well, we started with some HSP90 mutations in fruit flies. And this is an example, a wonderful example of how laboratories that create new tools and new materials can sometimes really drive forward the progress of science by being generous with their materials and sharing them with other laboratories. So Jerry Rubin and Ernst Hoffman's laboratory had both found HSP90 mutations in fruit flies. And they found them because they were interested in studying these two different, different signal transduction pathways in different contexts in the fruit flies that drove developmental processes. And not surprisingly, given what I've told you, mutations in HSP90 can affect those developmental processes in those pathways. But they weren't interested in HSP90 per se, so they were very, very generous, and they gave those mutations to us. What we found was, what they had found initially was that if the flies have, and of course they're diploid organisms, they have 2 copies of HSP90. If both of those copies of HSP90 are mutant, the organism die. HSP90 is a very important protein. They can't do without HSP90. If one of the copies is bad, they're pretty much fine, except that we noticed that they were only mostly normal, and that what we found was that a small number of the flies, 1 or 2% of the flies, had all sorts of different phenotypes. I mean, hundreds of different phenotypes. They had eyes that were growing off on stalks. They had bristles in the wrong places. They had wings that had notches in them. They had just all kinds of crazy phenotypes. And I had at this time been working in Drosophila for a while, and I'd never seen the effects of a mutation have so many pleiotropic effects and create so many different phenotypes in the fly, especially when only 1 copy of the protein was mutated. So we get new traits when HSP90 is inhibited, and here's even more examples of new traits. And what we found was that HSP90 inhibition, so this was genetic inhibition of HSP90, 1 mutated copy of HSP90, so you've reduced the buffer, you've reduced the excess amount of HSP90. That compromise of HSP90 was revealing the effects of hidden genetic variation. So it wasn't just destabilizing development in a random way. And the way we found that out was that it turns out that in different strains of fruit flies, when we crossed those same HSP90 mutations into different strains of fruit flies, we got completely different phenotypes in those strains because there was genetic variation in those strains that affected what HSP90 was able to do. We also found that in addition to genetic experiments, one way of really, this was kind of a crazy result at first, right? And so we wanted to be sure that it was due to HSP90 inhibition. And so we used a variety of tools. I showed you the genetic one. We also used those pharmacological tools to inhibit the HSP90 buffer, got the same effects. And then we said, well, again, maybe this is got something to do with, is a way of connecting environmental variation to the appearance of new phenotypes. So we asked whether simply growing the flies at higher temperatures would reveal those same phenotypes, those same new traits. And indeed, when protein homeostasis buffers were compromised by higher temperatures, we saw the same traits. The cool thing was that those traits, because they depend upon pre-existing genetic variation, the uncovering of pre-existing genetic variation, they can be assimilated by reassortment of that genetic variation in the next generations. So let me show you how this works in a simple cartoon form. We start with a population of flies that have a lot of different genetic variation in them that might contribute to a change in an eye phenotype. And, but none of the flies have that eye phenotype. You don't see anything. But when you compromise the buffer of HSP90, now a small number of those flies, 1%, as I said, or 0.5%, or sometimes 2%, but it was only a small number of the flies, suddenly saw a new phenotype. And then what we did was we mated the two different flies from lots and lots of bottles of flies. We mated pairs that had the same phenotype together. And we, of course, now understand that what was happening was we were selecting individuals that happened to have quite a bit of this genetic variation that would cause a change in the eye morphology when the HSP90 buffer was reduced. And in that very first generation, now instead of being in about 1% of the flies, it got up to 2 or 3%. And then the next generation, when we did that cross, it got up to 6 or 7%. The next generation, it got up to 10 or 12%. It took a long time, but after several generations of selective breeding, the flies in the subsequent generations had enough of that genetic variation that they had that eye phenotype, whether they were grown at high temperatures, whether they had the HSP90 mutation, or whether they were pharmacologically inhibited. It didn't matter. This trait, which initially appeared because of a compromise of the protein folding buffer in response to environmental stress, became fixed in subsequent generations by the recombination of that genetic variation to create a robust new phenotype. So then we decided to go back to yeast, basically. The reason for wanting to go back to yeast was because this organism, as I mentioned before, can be manipulated genetically better than any other organism on the planet right now. And that's actually due to the fact that beer brewers many, many years ago wanted to make better beer. So they started to manipulate the genetics of yeast. And it's just become a phenomenally important and powerful model organism. Now, we wondered whether in naturally occurring yeast strains from all sorts of different environments, we might see how broadly this effect of the HSP90 buffer is on the manifestation of genetic variation. So we were able to get strains from all sorts of different environments and all sorts of parts of the world due to the hard work of a large number of ecologists and evolutionary biologists who'd isolated these strains from the wild, taking great care to not manipulate them in the laboratory, but just get them put away right away. So we took these strains and we tested them for changes in growth phenotype. And so these are just, I'm keeping this pretty simple here, but we're growing the cells in this plate in one condition, one type of growth media, and over there we're growing them in a different type of growth media. And you can see some of the wells are light and some of the wells are dark. The reason for that is that we've got these plates sitting on a light box and we're shining the light through it. And so when the cells have grown, the light doesn't come through. The media is turbid. And the light shines through very, very nicely when the cells are not growing. And you can see that these strains are arrayed in both plates in exactly the same way. And you can see that the strains, some strains can grow in one media, some strains can grow in the other, some strains can't grow in either, some strains can grow in both. Now look at these strains here. These are 76 very different strains that we tested. Now we do something to inhibit HSP90. We add a low level of that HSP90 inhibitor that doesn't affect the growth of most strains under most conditions, and certainly not under conditions of rich media. But in these different conditions, we'd use different carbon sources, different nitrogen sources, all sorts of different conditions, high pH, low pH. What we found was that when we duplicated the growth curves under in exactly the same strains, exactly the same media, but in this case, we just have a little bit of HSP90 inhibitor in this plate. You can see that strains that were unable to grow before had genetic variation in them that allowed them a new trait to appear, allowed them to acquire a new capacity to grow when the HSP90 buffer was inhibited. And equally over here, you can see a whole bunch of traits that disappear. Those cells could grow perfectly well under that condition until the HSP90 buffer was inhibited. So these are examples in wild yeast strains where HSP90 is acting very broadly as a potentiator of some genetic variation and as a capacitor of other genetic variation. A potentiator that allows new traits that disappear when stress occurs, or as a capacitor which kind of keeps genetic variation hidden and then causes new traits when the HSP90 buffer is brought down. Now, most of these very same traits occurred in the absence of HSP90 inhibition when the strains were simply grown at higher temperatures. So again, a very natural environmental stress that creates a protein folding stress, uses up creates a lot of variety of protein folding stresses, but most particularly affects the HSP90 buffer, this protein which is normally there to serve in excess and depletes it. And the reason why we think that HSP90 is particularly important, again in this particular case, is that we're seeing the same kinds of traits with this environmental stress as we see with this very selective inhibition of HSP90. So yeast then allows us to sort of get some molecular understanding of how these traits occur. So we took two strains of yeast that had been isolated from very different environments, from grapes over here and from figs over there. And I've just kept this diagram very, very simple and pretended that the chromosomes of one strain are blue and the chromosomes of the other strain are red. We cross the strains. And then in the next generation, what happens is there's genetic reassortment of those genes through crossovers and recombinations and just assortment of the chromosomes. And you can see that the genetic variation has gotten mixed up in the progeny. And so what we could do, we could look at hundreds of these progeny and look for progeny where that shared the same genetic variation. So for example, this one has this little red region of the chromosome there, and that one over there also has in common the little red region of the chromosome. And so that allowed us to sort of map which regions of chromosomes, which regions of the genome were responsible for those traits. We actually mapped 400 traits. These are called quantitative trait loci because they could be measured in a quantitative way at how they affected growth. And we looked at traits across 100 different environmental conditions. And we found that fully 50 of them disappeared when HSP90 was inhibited, that buffer was reduced, and 50 new traits appeared when the buffer was reduced. So that's 100 of the traits that we had mapped depended upon, their manifestation of those traits depended upon this HSP90 protein folding buffer. And so we also were able to ask them with that experiment whether we could see evidence that this process had been really operating and had left an imprint on the genome sequences of these strains that exist today. So what we did was to simply, after having measured their growth rates under 100 different conditions, we simply lined up the strains according to, and these strains, sorry, I should have mentioned, these strains were sequenced by other laboratories. So we had the full genome sequence of these strains. And when you lined up the strains according to their genome sequence, you could see that the wine strains were quite closely related to each other. The clinical strains that we're looking at here were more closely related to each other. And that sake strain over there from Japan was really quite distant. And when we clustered by phenotype, that is, arranged them next to each other depending upon how similar they were under those 100 different growth conditions, surprisingly, it didn't line up with the genome sequence very well until we stressed them by reducing the HSP90 buffer or by simply exposing them to high temperatures. So what that really means is that there's a vast amount of genetic variation out there that would appear to be neutral and just random and chaotic and have nothing to do with the phenotypes of the strains until you stress the organism. So when you're growing the organism in the laboratory, you're missing a lot of the really important genetic variation that evolution has been acting on in those strains. And that's why the genotype-phenotype relationships match up much better when you inhibit HSP90. So can we see any evidence of this operating in fish or some higher organism? The reason why we turned to fish was because it's been a very, very special evolutionary system. And we had a wonderful, wonderful collaboration with Nick Roner and Cliff Tabin on this. And really, the work I'm going to be showing you in the next few slides was all their work. We just really helped as cheerleaders to get them going on the experiment and to really provide some advice and some hints about how to do things. So what they had is this very special system of evolution in which closely related fish that are growing out in the open waters have repeatedly been sequestered into caves. And in those caves, they evolve new traits. They for example, lose their eyes. And it's thought to be a selective advantage to lose your eyes, because if your eyes aren't doing you any good and you're bumping into things in the dark, they just provide a vulnerability for infection, etc. And they require a lot of energy to maintain eyes. So these are these surface fish and cave fish. And what we did was simply to use those inhibitors I told you about a little while ago to bring down the HSP90 buffer, and asked whether these are the surface fish, would we see hidden genetic variation in them that would cause the distribution of different types of eye sizes. And sure enough, and after they were treated with HSP90 inhibitors, sometimes the eyes got bigger and sometimes the eyes got smaller. What could have been the environmental stress that might be operating on this in these cave fish? If you consider their environment, it turns out that they're growing at quite similar temperatures, but in this case, they're exposed to different types of environmental stresses. They're growing under very different pHs with different amounts of dissolved oxygen and with very different amounts of dissolved solvents in the water that they're living in. So we asked whether those kinds of stress conditions, those environmental stresses, could reveal the same kinds of variation in eye morphology, and got very similar results. And then we had to ask, of course, whether that was really due to just randomized effects on development, or was it that it was due to underlying genetic variation that could be enriched in subsequent generations to start driving these fish towards a new phenotype. And so when Nick crossed fish together that had the smaller eyes after the HSP90 buffer was compromised, what he found in the next generation was that most of the fish had smaller eye size, even when the HSP90 buffer was not compromised. So it was in fact due to underlying genetic variation, and that genetic variation was enriched in subsequent generations. It could manifest as a smaller eye now without any need for HSP90 inhibition. And in fact, there was evidence that this has been happening with this cave fish evolution, because when we looked at the ability of these HSP90 inhibitors to cause variation in eye size in the cave fish, it did cause variation in eye size, but not towards larger eyes. They'd lost that variation under the selective pressures of living in the cave. So that was an example of a really wonderful collaboration with some really extraordinary scientists who are working on a model system that we couldn't begin to approach ourselves, and really excited about their willingness to try these things out. I just want to present maybe one more aspect of HSP90 and evolution in human beings. So this is just going to be one quick slide. This is a map of the human kinases and how they're related to each other. Kinases, as I mentioned earlier, are these signal transducers that do all kinds of regulate all kinds of processes in the cell. And we have systematically quantified the dependence of these different kinases on HSP90. And what we have found is that some of those kinases don't depend upon HSP90 at all. Others depend upon HSP90 moderately. And some of them are really strongly dependent upon HSP90. So you can see in very closely related kinases that they're acquiring mutations that contribute to their new functions, we think. But those mutations also cause them, like they did with the oncogenic mutations, to have a very differing level of dependence upon HSP90. So this is kind of an imprint of that HSP90 influencing the evolution of kinases in the human genome. So HSP90 transforms the adaptive value of large amounts of standing genetic variation. It affects polymorphisms that are located throughout the genome, and it even actually affects non-coding polymorphisms, and you can look at Dan Jarosz's paper for that. But what it does is does it in a combinatorial way, because this protein folding buffer stress means that lots of different mutations all of a sudden can exert their effects. So can create quite complicated phenotypes in a single step. And the traits can be assimilated by selection. So the like the drug resistance case, new mutations cause those resistant phenotypes that initially depended upon HSP90 to be more robust and to be present even when HSP90 was the inhibitor was there. And with the fruit flies, the selection of pre-existing genetic variation by reassortment in subsequent generations could also fix the traits. Simple environmental stresses exert the very similar effects on genetic variation. And HSP90 has sculpted then the standing genetic variation that exists in genomes today quite broadly. And understanding this aspect of human evolution matters importantly to human health. I've told you about just 2 examples of that. I've told you about drug resistance and I've told you about cancers, but there are many other examples as well. And there are many other features of this HSP90 buffer that I haven't told you about, that are work of other laboratories that have shown other ways in which HSP90 can influence the appearance of new traits. So HSP90 really provides a very plausible mechanism by which traits that are initially caused by environmental change can be assimilated and lead to new robust evolutionary traits. And one reason I've shown this picture of this same exact place 2 different times of year is just to remind you that this is really, these changes in environment are something that all organisms have been facing since the dawn of time. So it's not really all that surprising that HSP90 and many other mechanisms would be tied to the functionality of proteins tied to these effects of environment would actually be able to lead to new traits. And what's interesting about it is that this driving force for the evolution of new traits is not linked to the mutations themselves that cause the new traits. And that's one of the reasons why the system can survive. We're not saying that the system evolved in the first place to create evolution. We're saying that these proteins evolved to help other proteins fold. But in that process, the very fact that they help other proteins fold and that it exists in excess and can be used up by stress is what creates the consequence of that it also influences evolutionary processes. So with regard to Lamarck, who's been excoriated in the literature and in our textbooks for a couple of centuries now, I think the inheritance of environmentally acquired traits is a perfectly reasonable thing to contemplate and has happened, in fact, in evolution again and again. And it's time to give him back some of his dignity. And I want to finish by doing just one thing, and that is acknowledging the key players who did all of this work. I've been running the laboratory, but I haven't been doing experiments myself for quite a few years. And I have, however, been fortunate to be associated with these really remarkable people. Many, many other remarkable young people in my laboratory have contributed as well, but the particular experiments I showed you were performed by these people, and I want to give them all the credit. So thank you for listening.